Alright folks, it's 12 o'clock. Let's get started. So welcome back. I have a question for all of you. How was the exam yesterday? Was it easier than expected, harder than expected? What? Nothing. Worse than expected? So you don't have to yell your thoughts out in front of the entire class, but I'm asking you this so that you can think about what could be different moving forward, and you can give me feedback either through email or in person. And we'll have the grading done, I believe, by the end of this week, and then we can look for ways to make sure that you're better supported and equipped for the upcoming exams. Because I know that moving forward, the material will get extremely more challenging. Right? So let's get started with today's discussion. We'll be talking about the connective tissue, and I have a couple of questions for you before we get started. We're going to review what we discussed yesterday on different types of cell junctions. So if you were to have cardiac arrhythmias, which would be the malfunction of which of the following cell junctions could most likely lead to cardiac arrhythmia? Would it be a dysfunction of desmosomes, adherence junctions, tight junctions, hemidesmosomes? None of the above. Alright, we have like 40 plus responses. Okay, 12. So, most of the class said desmosomes, most said tight junctions, some said desmosomes, and only 4 of you said none of the above. And the 4 of you are right. The reason being, alright, I'm sure the rest want to know why did the 4 get it right, and I'll tell you. So speaking to the remaining 53 of you who did respond and the rest who did not respond. So we talked about gap junctions yesterday. And gap junctions were the kind of cell junctions which allow ions to move from one cell to the other. And that is very necessary for the functioning of the heart. You need to have that electrical current passing through in order for the heart muscle to contract and relax. And so because I did not mention gap junctions in any of the options, the right answer is none of the above. Is that clear? Alright, thank you. Since I saw at least 2 people nod, I'll move ahead. Mutations in claudins will most likely disrupt the structure of which of the following? So I told you that these cell junctions are made up of different types of proteins. There are hundreds of proteins, but I mentioned a few of them, and I discussed in a little bit of detail, so I'm hoping you can tell me if claudins had mutations, which of the following cell junctions would be disrupted? Okay. Ooh, most of you said tight junctions, and you are right. So, to answer this question, if you just remembered what we said in this slide, I told you that among tight junctions, there are a couple of transmembrane proteins, and only tight junctions have claudins, right? And so, if you knew this, you could have answered this question. All right. Now let's have a question on the epithelial cells. So, which of the following tissues has a protective function? Is it A, B, C, D, or E? Okay. Well, most of you said B, and B is right. The reason being, notice that B is stratified squamous epithelia. You see that there are multiple layers of cells at the apical surface, and that has a protective function. Whereas the rest have sort of a secretory function, or they have diffusion and absorption. So, I want to spend a few minutes talking about how you can answer questions based on histology in the future, right? So, there are three things I would tell you to look at whenever you have a histological slide. First is look at the nucleus. What is the shape of the nucleus? And is it flattened? Is it elongated? And then look at what is surrounding the nucleus. Can you see a bit of cytoplasm or not? And if the cytoplasm is sparse or if it's extensive, it's going to be helpful to know what tissue you're looking at. And also look at the apical surface as well. Hey folks, if you want to talk, could you leave the class and discuss, because I find it distracting. So I really appreciate it if we didn't have conversations happening in class. Okay, coming back to the apical surface. The apical surface is the part of the cell which is exposed to the outside world, or it's exposed to the cavity or the lumen, what you see here. And I also want you to look at the nuclei to see the number of layers of cells. So if you see here, you see that there are 2 layers of nuclei, so that indicates that we have stratified cuboidal epithelium, because you have 2 layers of cells, so it's stratified. And pseudostratified is the one case where you have the nuclei scattered all over, and because they're not in a neat line, we know that it is not multiple layers of cells, but it's just a single layer of cells. But given the specific slice you're looking at in the histological specimen, it looks like it's multiple layers, but it is not. And these 2 are also stratified epithelia, and this is the one which is from the skin, which I told you has a protective function. And transitional epithelia is one of its kind. It's the only one where you have a dome-shaped apical surface, and this is found only in the urinary bladder. So we will look at more examples of how to identify histological tissue in the upcoming lectures, but I just wanted to give you a primer of how to look at these slides when you do have an opportunity to. So what I will do first is we're going to wrap up with a couple of points which I did not have a chance to finish yesterday, and those are the characteristic of epithelial tissue. The first is that epithelial tissue, they have specialized contacts between each other and with the extracellular matrix, and we talked about this all of yesterday. We talked about tight junctions, which are towards the apical surface, followed by adherence junctions, desmosomes. Notice that adherent junctions and desmosomes, they help the cells connect laterally to each other, whereas hemidesmosomes and also focal adhesions will help the cell connect to the basement membrane. The second thing is that all these epithelial cells have polarity, and I told you that polarity is referring to the asymmetric nature of the cell, and the asymmetry could relate to the shape of the cell, where you have one side is an apical surface, the opposite is the basal surface. And notice that it's not only the shape, but also the function of the apical surface will be different than the function of the basal surface as well. The third thing you'll notice is that epithelial tissue, they're all avascular, meaning that they don't have blood vessels in them. And tomorrow we'll talk about the layers of the skin. And you see here, the upper layer of the skin, called the epidermis, is peeled off. This layer which is peeled off is made up of epithelial tissue, specifically stratified squamous epithelial tissue. And right below that, at this junction, you see that there are blood vessels, but those blood vessels are not going all the way to the epithelial tissue. So the only way nutrients or waste can diffuse to the upper layers is through diffusion, because the blood vessels don't innervate that part. And you will notice that there are a number of nerves which do innervate this epithelial tissue, and that's why the various senses of your body, like smell or touch, all of those signals can be sent to the brain. And lastly, we see that the epithelial tissue has a pretty short lifespan, and so they get regenerated pretty quickly. So whether if you have an upset stomach, usually gets recovered in a couple of days, because those cells get regenerated. If you have an injury on your skin, that also heals pretty quickly, and we'll see how that happens in when we discuss the layers of the skin. So with that, I complete with the material based on the epithelial tissue, and we move on to the new material for today. So I showed you this flowchart at the beginning, not at the beginning, I showed it to you on the first day of class and probably scared you all. I told you that we'll only look at a few examples, but guess what, I lied. We're going to look at each and every one example. And if you're scared by this picture right now, I just want you to know that you will not be scared by it by the time we get to the end of today's lecture. Because I'm going to give you some few tips on how to keep this information straight in your head. So when you think about the different kinds of epithelial tissue and how they are classified, I want you to keep two things in mind. The first thing is, all of these tissues, they would have different types of cells, right? So if you know what these cells are, you can tell what tissue you're looking at. And secondly, these cells are going to be surrounded by a matrix. And depending on the differences in the matrix, you could have different subdivisions, right? So different types of cells and the matrix surrounding those cells. Now there are two things which make up these matrix. One is the ground substance, which looks clear, and the other one are different types of fibers which you can actually see in this matrix, right? So we will see what makes up this ground substance, what are these fibers made of. So once you're able to know the different cell types and what the matrix is made up of, you should be able to tell why one connective tissue is different from the other. And not only that, you can tell the structures differently and you can say what are their different functions as well. Alright, so with that, let's jump right into the classification of connective tissue. So we can classify it based on as three types. One is connective tissue proper, followed by supportive connective tissue, and lastly fluid connective tissue. So this is the question I would first ask. When you think about fluids in the body, what is the first thing that comes to your mind? Blood, yes. Thank you. I was hoping you would say blood, and someone did say blood. So blood is a type of fluid connective tissue, but not only blood, but also lymph, which is made from the blood plasma. So that is also fluid connective tissue. When you think about what makes your body rigid and firm and supportive, what comes to your mind? Bones. Fantastic. Yeah. So we have bones are one type of supportive connective tissue, but we also have cartilage, which is found associated with these bones, and one function is it helps the bones from not rubbing with each other, and that's the function of cartilage. And so when you think about cartilage, we have three subdivisions. And then we finally move to the connective tissue proper, which has two major subdivisions, loose versus dense connective tissue. And within loose you have three subdivisions, and within dense you have three more subdivisions. And so we will walk through each of these, and hopefully by the end of today's lecture, this slide will not be scary anymore. So let's start with areolar connective tissue, which falls under the loose connective tissue. Now, the reason I like to start with areolar connective tissue is that it is the connective tissue which is most directly associated with the epithelial cell membranes, and we talked about epithelia yesterday. So if you take a look at this slide, you have simple columnar epithelia, and right below that you will have areolar connective tissue. And then here you have stratified transitional epithelia. Below that you have this areolar connective tissue. So here is a slide of what this connective tissue looks like. You'll notice that it is very loosely arranged, and there are cells, there's fibers, there's the matrix, but it's all held together very loosely. And you will find this is going to fill the spaces between muscle fibers, between blood and lymph vessels, and so on. And it's also going to be found in the abdominal cavity. So this is also a picture of the areolar connective tissue, and you can notice that there's a lot going on here, right? And there are a number of cells, there's different kinds of fibers, and it's a little hard to explain it using this picture, so I will use two schematic representations instead, right? So we'll use these two schematic representations, and my goal is to talk about each one of the things which are labeled on these two pictures and it'll hopefully all make sense. So when you think about the areolar connective tissue, it's a good place to start, because the various types of cells which are found in this will be found most likely in other connective tissue as well. So if you have a firm understanding of what the areolar connective tissue is, then you can easily understand the rest. So it's like one tissue to rule them all, right? So make sure you have a firm understanding of this areolar connective tissue. So let's start with the different types of fibers which are found in the areolar connective tissue. So I told you that the matrix is made up of two parts, ground substance and fibers, and we'll start with the fibers. So the first type of fiber I want to draw your attention to is the collagen fibers. Notice that these are straight and unbranched, as you can see here, and they're made up of fibrous proteins which give it a rope-like consistency. And even in this slide here, you notice that the collagen fibers are this lightly staining pink strands. So that is the collagen fibers. And the main function of collagen fibers is that they offer a lot of tensile strength, and they will resist stretching. So what gives your areolar connective tissue some sort of strength is this collagen fibers. And they help to hold the tissue together when there's different types of movement. Now, in contrast to the collagen fibers, you'll see that there are these darkly staining, very thin lines. So those are elastic fibers. And elastic fibers, they are not made up of fibrous protein, but they're made up of glycoproteins and especially elastin. That is the protein which gives elastic fibers their property of being stretched and coming back to their original shape. So note that the collagen fibers and elastic fibers, they have opposite functions. One is going to resist stretching, the other one is going to permit stretching and help the fiber to be elastic. Now the third fiber is called the reticulate fiber, and it's not very easily visible in the histological slide, and we will talk about this in more detail when we discuss reticular connective tissue. But you will see that these fibers are arranged in a branching network, and they will be found in tissues like liver, spleen, and it has a protective function. So now we are done discussing the three kinds of fibers, and we will jump into the different types of cells. So mesenchymal cells, they are multipotent adult stem cells which can differentiate into other kinds of cells based on if there is damage to the tissue. And the types of tissues, cells into which these stem cells can differentiate into would be fibroblasts, macrophages, and other connective tissue cells. So I told you that the cells can be of two types. One type of cells make the matrix, the other types of cells have an immune function, and that's exactly what fibroblast is. A fibroblast is a cell which is going to help make the matrix. And how it makes the matrix is it's going to secrete two types of things. One is polysaccharides, and the other one is it's going to secrete proteins as well. So the collagen protein which is secreted, the elastic fibers, all of that is made by the fibroblast cells. And what is being secreted is going to interact with water to form this viscous ground substance. And you will also find that these fibroblasts have a precursor cells known as fibrocytes. And I don't have a picture of fibrocytes, so I'm going to just use this schematic representation. So fibrocytes, they differ from fibroblasts in their shape. They're small and spindle shaped, whereas the fibroblasts are large and irregular shaped. And it is this fibrocyte, which is an inactive precursor, which will eventually turn into these fibroblasts. And the fibroblasts are what produce the extracellular matrix. So having discussed the one type of cell which makes the matrix, we'll talk about the other types of cells which are part of the immune system. So the first one type of immune system cell we'll discuss is macrophages. Right? So macrophage means a large eater, and we've talked about endocytosis, exocytosis, and these macrophages engage in phagocytosis where they engulf invading pathogens. And we did talk about macrophages very briefly yesterday when I talked about when the tight junctions are going to be impaired, then you have pathogens coming in, and that will be detected by these macrophages. And what these macrophages do is they will secrete cytokines. And I told you about interleukin 6, 23, and told you don't worry about it. We'll discuss it in the immune system. But yeah, I just want to point out that these are the cells which detect when pathogens come in, and they could either swallow up the pathogens or they can send signals to have other immune cells come and act on them. And you can have two types of macrophages, those which are fixed in the areolar connective tissue, and those are resident macrophages which act as the first line of defense. Or you could have field macrophages which can come in from the bloodstream to fight off the pathogens. So with that, I finish my explanation of this picture here, and we go on to the second picture. And in the second picture, I want to point out the second type of immune cell, and this is the mast cell. Notice that these mast cells have granules in them, and one type of granule being released is the histamines, and these are inflammatory mediators. So if you've had an allergic reaction and you want to take antihistamine medication, the antihistamine medication will basically prevent all this from happening. It's going to prevent swelling, redness, itching, inflammation, all of the things which the histamines are capable of bringing about. That's what an antihistamine drug would prevent. And I noticed that because there's going to be an increase in the blood flow, a second thing is also released, and that is heparin, which is an anticoagulant. And you want to minimize coagulation to enhance blood flow and to decrease the risk of clots. So these are the things released by the mast cells. So the other immune cells we'll consider are lymphocytes, which are going to be increased in their expression whenever there's some sort of damage. But these lymphocytes can also transform into plasma cells, which will produce antibodies against specific pathogens. And there are other immune cells you can also notice, like neutrophils and eosinophils, which kind of have a dumbbell-shaped nucleus, which we will talk about when we discuss blood. So these are called macrophages, which can engulf the pathogens as well. And the way these neutrophils and eosinophils work is that they're sent in response to the signaling from the macrophages and the mast cells. So with that, I complete my discussion on the different types of immune cells found in the areolar connective tissue, and we finally move to the ground substance. So we talked about the fibers, the different kinds of cells, and now we're coming to the ground substance. So the ground substance is made up of polysaccharides interacting with proteins, and the polysaccharides have a negative charge which will cause them to interact with water and draw the water in, and thereby making this viscous substance known as the ground substance. And this ground substance is very inflexible, and it is capable of occupying a large volume. And by occupying a large volume, it can also play a role in absorbing the shock experienced by the body due to trauma. And I will explain how this happens in a few slides, but first I want to just go on and explain what are the different types of substances which make up this extracellular matrix. And there are three kinds of macromolecules we will consider when we think about the matrix. The first one are the polysaccharides, also known as glycosaminoglycans, and we will look at their structure briefly. And then you have two kinds of proteins: fibrous proteins, which fall under the category of the collagen family, and non-fibrous proteins known as glycoproteins, which also are found. So this type of glycoproteins we will not discuss much in detail, so I just want to spend one slide talking about it, and then we won't be coming back to it. And an example of this glycoprotein is laminin, right? And let's see where this laminin can be found. So if you look at this schematic representation, you see there are three layers shown here. The uppermost layer is a simple squamous layer of cells. It is separated from the connective tissue by a basement membrane. And this basement membrane is shown very nicely in this electron micrograph. So what they've done is they've done some treatment to remove these epithelial cells, and so they've exposed the basement membrane. And below this basement membrane, you will have the connective tissue, which is the areolar connective tissue. See how loosely it is held together. And the basement membrane composition can vary from tissue to tissue, but the main protein found generally there is going to be laminin, which is the third type of protein which makes the matrix. So with that, let's look at the structure of the glycosaminoglycans. So these are polysaccharides which are unbranched, but they have these repeating disaccharide units. And there are two things I want you to take away when you think about these disaccharides. The first one is that they have an amino group, which makes sense, that's why it is glycosaminoglycans. And the second is that they have charged groups on them. So it could be either negatively charged carboxylic groups or sulfate groups. And it is this charge which leads to water molecules being strongly attracted to these polysaccharides and interacting with them. And if you remember what we discussed in lecture 2, we saw that the polar interactions between water and charged molecules is what helps them dissolve. And so we see that it is because of these charges, water is attracted and it causes the hydration of this extracellular matrix. So we see that these glycosaminoglycans can interact with proteins to form what is known as proteoglycans. And what you see here is an example of a proteoglycan called agrican. And the proteins part are shown as these two circles connected by these long cylindrical rod. And to this protein, you have a bunch of these glycosaminoglycans linked to it. And if you think about proteoglycans, 95% of it is going to be these polysaccharides, and only 5% is going to be the actual protein backbone. And these will be produced in the cells and released by exocytosis. But the question I have for you is, where do you think that these proteins will have the polysaccharides attached to them? In which type of organelle? Which organelle do you think? The ER, exactly, very good. It is in the ER. But the place where the elongation of these glycosaminoglycans takes place is going to be in the trans domain of the Golgi complex. Okay, here's the question you probably all are wondering about. How do glycosaminoglycans resist mechanical loads? And the way they do it, I will have to explain by using this type of representation. So this is also an agrican molecule, and I've just pointed out what are the different types of glycosaminoglycans. And in this cell, they're going to be hydrated because the water is attracted to the negative charge. But then when there is a force being applied on this area, we find that it's going to cause these proteoglycans to distort, and also it will cause the water molecules to be released. And this distortion and the releasing of water molecules will absorb the impact, and once that impact is taken away, the proteoglycans take back the normal position, but then the water can also flow down the concentration gradient, and then it starts to rehydrate these proteoglycans. So that's how they help resisting mechanical load. Is this clear? Alright. In that case, we are done with our discussion of the areolar connective tissue, and we should be wrapping up with the other things pretty quickly. I just have a couple of things to say about all of the other subdivisions. So adipose tissue is the second type of loose connective tissue, and it's made up of adipocytes. So that's the main kind of cell seen in adipose tissue. And adipose tissue, the adipocytes are also seen in the areolar connective tissue, but I didn't want to mention it back then because we're talking about it now. And notice that the fat which is inside these adipocytes, they're going to have a yellow tinge to it because fat-soluble keratin is dissolved and stored in the fat cells, and that's why they have a yellow tinge. But notice that there is also a lot of blood vessels which are closely associated with these adipocytes, and that's how the nutrients can be easily stored away as fat, and they can also be quickly mobilized again and sent back into the bloodstream. So that's how these adipocytes are going to be organized. And if you look at the way the histological slide, you'll notice that there are the nucleus is going to be pushed to one side of the cell because of the vacuole filled with fat, and so it has this chicken wire-like appearance. And notice that these fat droplets are going to stain very in a pale manner. So the way these adipocytes are going to be distributed through the body is mainly going to be subcutaneously, meaning under the skin. But there is also something known as the ectopic fat, which is going to be distributed in other parts of the body, as shown in this picture here. So the reason why sometimes fat is going to be distributed ectopically is because this subcutaneous capacity has been reached, and the way this ectopic distribution can really depend on individuals. And so I was interested in this study here, it's called a masala study, which is done on people of different ethnic groups living in the US. And so what they found was that compared with Chinese Americans, African Americans, and Hispanics, South Asians living in the US had greater visceral fat. So you may be wondering, why are you interested in South Asians? Because that's what I am. People from India or Pakistan, Bangladesh, Sri Lanka, they're all South Asians. And so it's an interesting study. The implications of this I'm happy to discuss with you after class and why this is the case. But I just wanted to use this example to say that how there is a genetic component in how fat can be distributed as well. And so we come to the last type of loose connective tissue, and this is reticular connective tissue. And the word reticulus means little net in Latin. And you see that these little mesh-like fibers are formed because of the fibroblastic reticular cells. And these kinds of meshes will allow other cells to attach on. And the purpose of this kind of tissue is to offer support to soft tissues like lymphatic tissue, spleen, and the liver. So that's it. Nothing more to say about this kind of tissue. And we move on to dense connective tissue. So the dense connective tissue can also have 3 subdivisions, and the regular dense connective tissue has the name because of the way in which the matrix is arranged. Notice that the matrix has got parallel layers of collagen fibers, and you know that these collagen fibers because of the light stain they have, and all of these dark lines you see alongside, those are the fibroblasts. And because they're very closely packed together, they have a wavy appearance, but it also makes this tissue extremely strong. And so you would find them in ligaments which connect bone to bone, or you would find them in tendons which connect muscle to the bone. And just to tell you how strong collagen is, if you were to look at steel of the same diameter and pull it lengthwise, collagen is definitely stronger. And so when you think about muscle contraction, if there is going to be an uncontrolled muscle contraction, it is more likely that the bone will break rather than the tendon or the ligament snapping it through. So that's the level of strength offered because of this dense connective tissue. Now, irregular connective tissue will also have collagen fibers, but notice that they are going to be arranged in a less organized way, and it is more random. Now, the advantage of this is that you will have the tissue can move in different directions with a reasonable amount of strength, but if you were to pull it very strongly in one direction, it would give way, but still it gives more flexibility in the directions with which you can move the tissue. So we know that muscles will contract in one direction, but a place where you need to have this type of flexibility is in the dermis, which is under your skin. So we will talk about the integumentary system and the layers of the skin tomorrow in great detail. But just note that the dermis has got two parts. The part which is closer to the epidermis or the epithelial cells will be made up of the areolar connective tissue, but the part below is made up of irregular dense connective tissue. Now the reason for this is that the part in the lower part of the dermis needs to support a number of appendages or structures like the hair follicles and sweat glands, and so that is the reason why it has to have a little more support and be firmer than the areolar connective tissue. Are there any questions at all at this point? Alright, it's just information from my mouth to your head at this point. And we look at elastic connective tissue. Now the thing is, this will be found in areas where there needs to be stretching, and then the shapes, the structure comes back to its original shape. Examples of this include bronchial tubes or vocal cords, and also the arteries and blood vessels. And what you see here is a picture of a human aorta magnified many hundred times, and these dark lines are the elastic fibers. And we did talk about the darkly stained elastic fibers when I mentioned areolar connective tissue, and here you see these are darkly stained. And the interesting thing is you will also be able to see smooth muscles in this area and the ground substance as well surrounding these elastic fibers. So with that, we move on to cartilage. And the interesting thing about cartilage is that it's going to be softer and weaker than bones, but the main function, it helps bones from not rubbing against each other. So the way the cartilage can protect bones is that it's made up of a unique matrix made up of proteoglycans and polysaccharides. But in this matrix, you have chondrocytes. Those are the cells which are specific to cartilages. We saw that in areolar connective tissue and dense connective tissue was fibroblasts. In adipocytes were in the adipose tissue, and then you have chondrocytes in cartilage. So we have these chondrocytes which are quite far from the blood vessels because these cartilage tissue is avascular. So any sort of damage or injury, it takes quite a while to heal. So when you think about the three types of cartilage, in all of these three types, you will notice that there are these chondrocytes in lacunae, but the main difference comes in the type of matrix which is surrounding these chondrocytes, and I want to highlight what are those differences in the matrix. So if you look at the hyaline cartilage, the matrix has a very hazy or a glassy appearance because you have primarily large amounts of proteoglycans, and you have some amount of collagen fibers which are dispersed. That's why it has this very light appearance. And these are strong and flexible, and so they're going to be found at the ends of long bones to prevent the bones from rubbing against each other. And when you have loss of this cartilage at the end of long bones, it will lead to a condition known as osteoarthritis. We also see hyaline cartilage is going to form the template of the embryonic skeleton. So what you see in this figure is you see how the bone is formed from the embryonic skeleton all the way into adulthood. And notice that this is a figure we can study in greater detail when we do skeletal system. But for now, just notice that there's hyaline cartilage which is forming the template. And as the bone develops and it gets calcified and has blood vessels being formed, you'll notice that there is this region called the epiphyseal plate. So the epiphyseal plate is also made up of hyaline cartilage, but then once growth stops and once the child has reached the height, this region will be replaced with bone as well. So in fibrous cartilage, we see that there's a lot more collagen in the matrix, and they are arranged in alternating rows. And as a result, this fibroelastic cartilage will be a lot more stronger than the hyaline cartilage, and it functions to prevent bones from compressing against each other. So you will see fibroelastic cartilage in intervertebral discs or between the pubic bones, and this is known as a pubic symphysis. So during labor and childbirth, this pubic symphysis can slightly be dislodged out of place as the baby is being born as well. And finally, we talk about elastic cartilage, and the elastic cartilage has fewer collagen fibers, but what it predominantly has is the darkly staining elastic fibers. And here is a picture showing you the elastic fibers which stain darkly, and one place where you would observe this would be the human ear because it's pretty elastic. And then we move on to bones. So we see that the bone structure can be divided into two parts. You have the cancellous or the spongy bone, which is towards the ends of the long bones, and you have the compact bone, which is in the middle. But regardless of their structure, if you look at the nanoscale architecture, you notice that the matrix is made up of a bunch of collagen fibers, and in these collagen fibers you have crystals, hydroxyapatite crystals, which are made up of calcium phosphate, which are found there. And both are important because if you were to take away the collagen, the bone will be super brittle and just shatter. But without the minerals, the bones will offer very little support. And so I want to point out what this would mean by showing you this quick video. So see what she's doing. She's she's put these chicken bones in vinegar for a couple of weeks, and then see what happens. Because the minerals are being dissolved by the vinegar, the bone is pretty rubbery, and you bend it like, yeah, it's rubber. Right. So when you look at the structure of a bone, a histological view, you see that the cross section reveals that there is the central canal which is surrounded by these darkly staining circles of lamellae. So we'll see what this is. The central canal is going to have spaces for blood vessels, lymphatic vessels, and also nerves. That's why if you break a bone, it's going to hurt a lot because there are nerves there. But also you notice that in the surrounding spaces or the lamellae, you have osteocytes, which are the cells specific to the bones. And there are different types of osteocytes, and we can look at that detail later when we discuss the skeletal system. But because of this intense vascularization, if there is some sort of damage which takes place to the bone, the recovery can be done completely. And then I just want to point out the difference between the spongy bone and the compact bone. So the spongy bone, it's got a lot of empty spaces, and that's why it is less dense in compared to the compact bone, which has got fewer spaces and it has greater structural strength as well. So finally, we come to blood and lymph under the fluid connective tissue. And we will be talking about the various cells in the blood and also the plasma in which the blood cells are found. But for now, I just want to focus on how lymph is made. So what you see here in this picture is the blood being circulated throughout the body, and as blood is being circulated, it's under a lot of pressure because of the pumping of the heart muscle. And this blood pressure is going to cause some of the plasma in the blood to move into the surrounding tissue or into the interstitial space. Now, most of this tissue in the interstitial space is going to be reabsorbed back into the blood vessels. But some of this doesn't go back to the blood vessels. In fact, it's going to enter the lymphatic system. And the interstitial fluid which enters the lymphatic system is the lymph. And we will be talking about the function of this lymphatic system and when we discuss the immune function. So with that, we discussed about the various components which make up the different types of connective tissue, and I want to highlight what are the functions of these various connective tissues. So the main function of the connective tissue proper is going to be to connect various organs and to protect them. In terms of the dense regular connective tissue, we see how bones and muscles can be connected. We also see how the areolar connective tissue can link the epithelial tissue to the underlying layers. And we see the fat can also have a role in protecting from trauma, and it can serve as a source of energy as well. In terms of supportive connective tissue, both cartilage and bones not only has structural strength, but it also gives the framework to the body. But when we move on to the fluid connective tissue, we see that the blood can circulate the dissolved materials like nutrients, oxygen, or carbon dioxide, or even waste products. And there's also a protective role from pathogens by the circulation of the white blood cells. So with that, I'd like to summarize what we've been discussing. I hope that this flowchart is no longer scary now that we have gone through the various subdivision of the connective tissue. So we saw that the subdivision was based on the types of cells found in these different types of connective tissue, and also the matrix which surrounds those cells. And depending on how tightly those matrix was connected, we would have either greater strength or more flexibility. And based on the structure of these various connective tissue, we see they have various functions which range from protection, support, energy reserves, tensile strength, flexibility, transport, and immunity as well. So with that, I end today. Thank you for your time, and I'll see you all tomorrow to start our discussion on the lowest exam. The lowest of the first 4 exams. The final exam can be dropped.
It can't.
Cannot. The final cannot be dropped.
But does the final replace the lowest exam, or is it just dropped?
They would drop the lowest exam. Yeah. And so the thing is, so the final exam will count for 200 points.
Okay. Normal exam is 100, right?
100 points. So your total will be out of 500 points.
Okay.
So if you have 4 main exams and you have 3 of those, 100 points each, and 200 for the final plus 500 points.
Okay. And then I feel like students did not do well on the first exam.
It depends.
I studied a lot, and I don't think I got like the grade I wanted.
Okay.
So I was just wondering like how people do this.
We
and
will,
like if like the like if it's one of those classes where it's like super hard, but you got extra credit to compensate it.
I really doubt. See, look at the performance, because I have a few students who have done much better than I would imagine. So I think we can have a discussion as the results are out, and we can also see what questions students mostly got wrong, and then accordingly. Yeah, so thanks.